is Kruti Patel from Pharmaceutical Technology Department, LJ Institute of Pharmacy. Welcome to Pharma Ignite, the official channel of LJ Institute of Pharmacy. Today we are going to continue with the sterilization only. In sterilization, we had seen the overview of sterilization methods and chemical sterilization methods. Today we are going to start with the physical methods of sterilization. So, quickly revising what the methods are included in physical sterilization. Then first is heat sterilization. In heat sterilization or high temperature, we have two different categories. First is dry heat sterilization, second is moist heat sterilization. In dry heat sterilization, we are going to learn about hot air sterilization. Then in moist heat sterilization, we are going to learn about steam under pressure, tyndallization and pasteurization. And the next method which we are going to learn today is radiation sterilization. Okay, so in today's video, in today's session, we are going to cover this many different methods. Let's start with the first, hot air sterilization. Now for the hot air sterilization, the instrument which we are going to use is oven. So this is the diagram of oven. The main mechanism of killing of microorganism is oxidation of microbial cell. This mechanism of killing of microorganism is important, that you need to remember. Okay, so yes, microorganisms will be killed in hot air oven by oxidation method. Okay. If we look at the advantages, then main advantage is it is suitable for the material that are damaged by moisture, for example, oils and powders. It is less damaging to glass and metal equipments than moist heat sterilization. It does not leave any chemical residues, and of course, that eliminates the problem of wet pack. If we look at the disadvantages, then first is rubber and plastic materials cannot be sterilized by this method. Preparations containing water, alcohol or volatile liquids cannot be sterilized as they may evaporate at high temperature, and not suitable for surgical dressings as natural moisture evaporate and lead to deterioration. Looking at the applications of hot air oven, it is used to sterilize glasswares like flasks, beakers, tubes, pipettes, petri dishes, glass syringes, then equipments like mortar and pestle, evaporating pans, scissors, scalpels, ointment tubes and surgical instruments, then also oils like fixed oils, ointment bases, liquid paraffin, wool, fat, wool, alcohol, beeswax and powders like talc, kaolin, starch, lactose and zinc oxide. The next method is steam under pressure. Now, I have drawn a table, right, which indicates pressure, temperature and time. As you all know, when we boil water, that can only achieve 100 degrees Celsius, right, and at 100 degrees Celsius, that means 0 pressure, 0 psi, that is pound per square inch, right. So, at 0 psi pressure, water will achieve only 100 degrees Celsius and the sterilization time which is required is 40 minutes. Okay. Now, if I apply pressure, if I apply 10 psi pressure, then the temperature which can be achievable is 115 degrees Celsius and the sterilization time which is reducing to 30 minutes. Okay. Now, this is official in British Pharmacopoeia, they use the standards. Okay. Now, again I am increasing pressure. Okay, I am using 15 psi pressure, then sterilization temperature or steam steam temperature which I can achieve is 121 degrees Celsius and the sterilization time again reduces to 15 minutes, right. Now, if I increase more pressure 20 psi, then the temperature which I can achieve is 126.5 degrees Celsius and sterilization time is still reducing, that is 10 minutes. Okay. But in USP, the official is 15 psi at 121 degrees Celsius for 15 minutes, okay, which this is official in USP, and we usually use this temperature, pressure and time combo for the sterilization by steam under pressure. Okay. So, if we look at the mechanism of killing of microorganism by steam under pressure, then it is denaturation and coagulation of bacterial proteins. Now, what happens at higher moisture content, the sulfhydryl groups will be released and they will be forming small peptide chains. Now, this small peptide chains, they will be forming new complex structure which is different from actual protein structure, and because of that microorganisms will be killed. Okay, so that will be the reason for the death of microorganisms, right? These are the diagrams of autoclave. Two types of autoclaves are available. First is vertical autoclave and second is horizontal autoclave. Looking at the advantages of autoclave, then penetration power of steam is high. So, microorganisms are killed more efficiently at higher temperature and lesser time. Then large quantity of materials can be sterilized and sealed containers and ampules are readily sterilized. Disadvantages, then it is not suitable for oils, ointments, powders and oily injections, difficult to produce dry packs, and plastic items cannot be sterilized. If you look at the applications, then it is used for the sterilization of screw caps with rubber liners, rubber closures, injection solutions like NaCl and dextrose, suspension and surgical dressings and fabrics can be sterilized, and also culture media and certain equipments can be sterilized using steam under pressure. Now, the next method is sterilization, or which is also known as fractional sterilization, right. Now, this method is official in BP. This is the method for the products which cannot withstand 115 degrees Celsius temperature, right. But if they can withstand 80 degrees Celsius temperature, then this method can be used, right. At 80 degrees Celsius, it is difficult to kill microorganism, right. So, what we will be doing, we will be performing this method for 3 successive days, 3 continuous days, okay. And you have to sterilize your articles for 1 hour at 80 degrees Celsius with in between incubation. That means for today, if you are sterilizing your product for 1 hour at 80 degrees Celsius, after sterilization you have to incubate them, and after 24 hours tomorrow at the same time you have to again perform the same method for the sterilization, okay. Then you have to incubate them, and for the 3rd day, that is the last day, again you have to perform sterilization. Why we are doing so? To kill spores, okay. Now, spores are very difficult part of very difficult microorganisms to kill, right. So, what we will be doing, we will be first exposing them to harsh condition. So, microorganisms will be converted into spores, right. After giving harsh conditions, we will be providing them temperature or conditions to grow. So, all spores will be converted to vegetative cells, right. Once they are converted into vegetative cells, we will be killing them. So, this we are repeating for 3 days, and it is said that by performing this method for 3 successive days, most of the microorganisms, that is 99.99% microorganisms can be killed, right. Okay, so the instrument which is used for this method is steam Arnold, which is very similar to your autoclave, right, or you can also use autoclave without using the pressure valve, okay. So, yes, this is fractional sterilization or tyndallization, right. The next method is pasteurization. Now, this method was developed by Louis Pasteur, right, that is why it is known as pasteurization. Now, this method was initially developed for to improve the storing property of wine, right, to preserve wine. This method was used, but later on this method is used for all products, all food products, and widely for the milk, right. So, if we look at the pasteurization, then that can be done by 2 main methods. First is Holder method, okay, which is performed at 62.8 degrees Celsius for 30 minutes. That means you have to heat your milk at 62.8 degrees Celsius for 30 minutes, and then you have to quickly cool it down, okay. The second method is high temperature short time method. Now, in this, the temperature is 71.6 degrees Celsius and the time which is used is 15 seconds. Please make sure it is 15 seconds only, right. So, you have to heat your milk at 71.6 degrees Celsius. When you achieve that temperature, you have to keep it only for 15 seconds, that is all, and then you have to quickly cool down, okay. So, yes, by this method what happens, your product is not much damaged, okay, and microorganisms will be killed. So, these are the 2 main methods by which you can perform pasteurization. A novel novel methods which are used for the pasteurization are, first is ultra high temperature. Now in this ultra high temperature, you have to heat your milk at 138 degrees Celsius. You have to keep it only for 2 seconds. 2 seconds, right? You have to keep your product only for 2 seconds, then you have to quickly cool it down, and then you have to pack it in, hermetically sealed containers, right? That means your 6-layer Tetra Paks, they are known as hermetically sealed containers, right? This is example of hermetically sealed container, right? So you have to pack them in hermetically sealed containers, and then you have to supply them, okay? So that is ultra high temperature. Second method is ultra pasteurization. In this method, again you have to heat your product at 138 degrees Celsius for 2 seconds only, but the product is not packed in hermetically sealed container. That is the only difference. Okay, so these two are the novel methods for the pasteurization. The next method is radiation method. Now, if we talk about radiations, that can be achieved by 2 different methods, or they are divided into 2 main categories. First is electromagnetic waves, which include IR, UV, right, X-rays, and second is stream of minute particles of matter, which includes alpha, beta and gamma particles. Now let's talk about first, that is electromagnetic waves. Now from electromagnetic waves, the sterilizing capacity is with UV rays, okay, which having the range of 200 to 300 nanometers, right. This range is useful for the sterilization capacity, from which the most efficient wavelength is 253.7 nanometers. Okay, so yes, UV can be used for sterilizing agent with 253.7 nanometers capacity, right. Now let's learn about the mechanism of killing of microorganisms by UV method. The microorganisms are killed by 2 different modes. First is direct action and second is indirect action. If you look at the direct action, then when UV falls on the microbes, it is absorbed by the DNA molecules. 2 thymine nearer to each other form bond. So thymine dimers are formed. This in turn is known as mutation, and due to this error, the body system tries to repair it by cutting the thymine dimers, which requires longer time, and by that time microorganisms are killed. Looking at the indirect action, sometimes some poisonous materials like hydrogen peroxide is formed due to action of radiation on some of the ingredients of medium, which act as cell poison, and finally that is killing the microorganisms. The main disadvantage of UV is its penetration power. The penetration power of UV is very less, right? Even it cannot penetrate through a thin layer of paper also. Okay, so that's why using UV limits our possibilities. Okay, otherwise it is excellent sterilizing agent. Next is ionizing radiations. Now ionizing radiations can be obtained by 2 methods. First is machine generated, okay, and second is radioactive isotopes. Now mainly the radioactive isotopes are used, and mostly alpha and beta particles are used for the sterilization purpose. Looking at the mechanism of killing of microorganism by ionizing radiations, then they act by 2 different modes. First is direct action, that means when ionizing radiations passes through cell nucleotides, they absorb that radiations and thus they get ionized and tends to separate, and finally whole DNA structure breaks and cell dies. Looking at the indirect action, microbial cell contains abundant amount of water. When radiation is absorbed by the water, it produces free radicals. These free radicals are powerful oxidizing and reducing agents capable of damaging essential molecules inside the cell structure and thereby death of microorganisms. I hope all the methods of physical sterilizations are clear to you. For more such videos, please subscribe Pharma Ignite. Thank you so much.